Hello students, welcome to biology class. In the chapter Microbes in Human Welfare, next we're going to discuss about microbes in sewage treatment. So here we're going to study about the two treatment, one is the primary and other one is the secondary treatment. So next we're going to study about the microbes in the production of biogas. So all of these we are going to cover in this video. Microbes in sewage treatment. What is sewage? Sewage is a municipal wastewater containing large quantities of human excreta. I repeat, sewage, it is the municipal wastewater containing large quantities of human excreta. A major component of wastewater that are generated every day in cities and towns in human excreta. So this municipal wastewater is called sewage. So this will be in other terms. Why do we need the sewage treatment? Because sewage contain large amount of organic matter and pathogenic microbes. Before discharging into natural bodies, sewage is made less polluting. So sewage is treated in sewage treatment plants. So in short, we will be calling that as STP. So STP is nothing but sewage treatment plants. So here in this slide, we're going to study about the STP. So what is STP? Sewage treatment plant. Before it is discharged into water bodies to reduce the pollution load, the treatment of wastewater is done by the heterotrophic microbe naturally present in the sewage. Ganga Action Plant and Yamuna Action Plant have been initiated by Ministry of Environment and Forest, under which large amount of STP will be built to treat sewage before they discharge in the river to reduce the pollution. How is the sewage treated using STP? So here it involves the two processes, that is the primary treatment and secondary treatment. To follow the STP, so here we are undergoing the two treatment, one is primary treatment and other one is secondary treatment. What is primary treatment? It is the physical removal of large and small particles from sewage. Whereas in secondary treatment, the primary effluent is passed into large aeration tank with constant mechanical agitation and air supply. So by this way, we are doing the secondary treatment. Primary treatment, it involves physical removal of particle that is large and small from the sewage through filtration and sedimentation. So here the process and the technique involved is filtration and sedimentation. Kindly make a note. These particles are removed in stages. So the removal of floating debris by sequential filtration is the first process. Second one, the grit removal by sedimentation in the primary settling tank. So these are the two stages, that is the filtration and sedimentation process done in the primary treatment. During the sedimentation, most organic and inorganic solid settled, so that we will be calling that as a primary sludge, while floating materials like grease and plastic rise to the surface called primary effluent. So here we have a question, what is the difference between primary sludge and primary effluent? So primary sludge is nothing but the sedimented particle, that is the organic and inorganic solid which is settled down, is called primary sludge. Whereas primary effluent is nothing but the floating materials like a grease and the plastic rise to the surface when we doing this primary treatment. I repeat the point again for the primary treatment. It is a physical removal of large and small particles from sewage. First, the floating debris is removed by sequential filtration by passing through the wire mesh screens. Then the grit. The grit is nothing but the soil and the small pebbles are removed by sedimentation in settling tanks. So the sediment is called primary sludge and supernatant is the effluent. So here we studied what is the difference between primary sludge and the primary effluent. So this effluent will be taken for the secondary treatment. So once the process primary treatment gets over, next we move on to the secondary treatment. So secondary treatment process will be moving on with the primary effluent, that is the floating materials like grease and plastic rice. In this picture, you can study the difference between the two processes. One is filtration and other one is sedimentation. See, filtration is nothing but you're filtrating. Okay, that's it. Sedimentation is the grit which will be like removed where it will be sediment in the tank. So, first picture shows the removal of debris through filtration. Second one shows grit removal in primary settling tank through sedimentation process. So next we're going to discuss about the secondary treatment. So secondary treatment is nothing but, in other term we will be calling as biological treatment. That is, we were using some microbes to be in this treatment. The primary effluents are passed into large aeration tank. So what is the primary effluent? That is the floating material. The main aim of the secondary treatment is to reduce the organic matter in effluent. In the tank, the effluent are constantly agitated mechanically and oxygen is pumped into it. As a result, the aerobic bacteria naturally present in the effluent grows into floc. What is floc? Flocs are mesh-like structure formed by group of bacteria in association with fungal filaments. In simple words, secondary treatment is passed into a large aeration tank with constant mechanical agitation and air supply. So with the help of the useful aerobic microbes, grows rapidly and form a flocs. So these flocs are a mass of the bacteria, okay, or the masses of bacteria associated with fungal filaments to form a mesh-like structures. So these growing microbes consume the organic matter and thus reduce the biochemical oxygen demand, that is BOD. So now we're going to discuss more about BOD. What is BOD? Biochemical oxygen demand. In the presence of oxygen, these growing aerobic microbes consumes the organic matter present in the effluent. The secondary treatment reduces the BOD of water. So what is the definition of biochemical oxygen demand? It refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. Okay, in other term, it refers to the amount of oxygen consumed if all the organic matter in 1 liter of water is oxidized by bacteria. BOD measures the amount of organic matter in water by measuring the rate of oxygen uptake by microbes. Higher BOD indicates the higher polluting potential. See, in water pollution, with the help of this BOD level, that is the biological oxygen demand itself, we yet to know how the water is polluted. Whether it is a higher polluting agent or the lower polluting agent, we can be able to know the potential of the pollutants. So higher BOD level, it indicates the higher polluting potential. So here in this slide, you can able to read about the difference between the BOD in different snaps. So the BOD of drinking water should be less than 0.5 mg per liter. However, the BOD of raw sewage can be as high as 600 mg per liter. The greater the BOD of sewage water, I said before, it will be what? It will be the pollution indication. So the more polluted it is. Secondary treatment is continued till BOD decreases considerably. Once the BOD reduces, the effluent are transferred to another aeration tank where the flocks of bacteria are gradually allowed to the sediment. So this sediment part of sewage is known as activated sludge. See, when BOD of sewage has reduced, the effluent is passed into the settling tank. Here, the bacteria flock settle and the sediment is called activated sludge. The activated sludge are pumped into large tank called anaerobic sludge digester. See here, In the right part of the slide, there will be the aeration tank. So that is an anaerobic sludge digester. So what does this digester will do? Yes. So here anaerobic bacteria will digest the bacteria and fungi present in the activated sludge and produce a mixture of gases such as methane, hydrogen sulfide, carbon dioxide. See, whenever you be opening this sewage tank, so there will be like the smell, right? So that smell is only because of the hydrogen sulfide gas which is present into that. So this mixture of gases is called biogas. So what is biogas? The mixture of the gases like methane, hydrogen sulfide, carbon dioxide. So biogas is used as a fuel since it is inflammable. Methane is the main gas produced during sewage treatment and also serve as fuel. Methane is used to produce heat in homes and factories. The sludge left after digestion, about the aerobic and anaerobic bacteria, is used as a manure and it is called activated sludge. So the effluent now remain from the secondary treatment plants are now fit to released into natural water bodies like rivers and streams. A small part of the sludge is used as an inoculum in the aeration tank and the remaining part is passed into the large tank called anaerobic sludge digester. So in this digester, the heterotrophic microbes anaerobically digest the bacteria and fungi. So all the digestion process, once it's open, what happened, the methane, hydrogen sulfide gas and carbon dioxide gas will be released. So that form the biogas. So this will be the final part of the secondary treatment where the effluent now remain from the secondary treatment plants are now fit to release into the natural water bodies like rivers and streams. However, a small part of the activated sludge is pumped back into the aeration tank to save us inoculum for subsequent treatment. Due to increasing urbanization, sewage is being produced in larger number than before. However, the number of sewage treatment plant has not increased enough to treat such large quantities. So, the untreated sewage is often discharged directly into river, leading to pollution and increase in waterborne diseases. So, this flow chart shows the microbial treatment of sewage. Okay, so this will be like a recap session. See, the primary treatment and secondary treatment, they are the two types of the process which is involved in microbial treatment of sewage. In primary treatment, it meant for only the physical one. Secondary treatment is meant for the biological process. So in primary treatment, it involves a primary settling tank, filtration and sedimentation process where we studied here. So this primary treatment, the result will be the primary sludge will be carried over to the secondary treatment with help of the inside aeration tank, the BOD reduced by the flock. A part of activated sludge sent back to the inoculum. Inside the second aeration tank, the flock sediment called activated sludge. Inside the anaerobic sludge, digester microbial activity will be doing. At the end product of the secondary treatment, so there will be like production of biogas, effluent can be released into river and stream, and manure will be getting. So these are microbial treatment of sewage. Now we are going to discuss about microbes in production of biogas. Microbe, okay, will be used in the production of biogas. So what is biogas? Biogas is a mixture of inflammable gases, methane, SO2, CO2, etc. which is produced by a microbial activity that can be used as a fuel. So here the bacteria involved called methanobacterium. Okay, so that we will be calling as the methanogens. Anaerobically breaks down celluloid material to produce CO2 and H2N. Anaerobic sludge in several sewage treatment plants, the rumen of cattle, thus providing the nutrition to cattle. Okay, see, IARI, Indian Agricultural Research Institute and Khadi and Village Industries Commission KVIC developed the technology of biogas in India. So this picture shows the mechanism of biogas or gobar gas production. See, the biogas is otherwise called as gobar gas. The raw material for biogas production is excreta, that is the dung of a cattle. The biogas plant has a concrete tank 10 to 15 feet deep in which bio-waste and slurry of dung is collected. So the tank has a floating cover which rides on production of gas in the tank. Methanobacterium in the dung act on the bio-waste to produce biogas. The biogas produced is supplied to nearby house by an outlet. Through another outlet, the spent slurry is removed to be used as a fertilizer. So biogas is used as a fuel for cooking and lighting. So next topic we're going to discuss about microbes as biofertilizer. Biofertilizer are the microorganisms which enrich the nutrient. Okay, so that is the highlight point of biofertilizer, where it has a nutrient content like nitrogen, phosphorus, etc. So bacteria, fungi and cyanobacteria are the three main sources of biofertilizer. So first we're going to study about the bacteria as biofertilizer. So the examples are Rhizobium, Azospirillum and Azotobacter. So these are the bacteria, okay, which have the enrich of the soil nutrients. Okay, it helps to enrich the soil nutrients. Actually, the Rhizobium is a symbiotic bacterium that lives in the root nodule of legume and fixes the atmospheric nitrogen into organic compound. So next is about fungi as biofertilizer. Fungi form the symbiotic association with the root of higher plant called mycorrhizae. Example, glomus. The fungal hyphae absorb phosphorus from the soil and passes into the plant. Next, cyanobacteria as biofertilizer. Cyanobacteria is blue-green algae. It helps to fix the atmospheric nitrogen and increase the organic matter of the soil through the photosynthetic activity. Example, nostoc, hanabina, oscillatoria, etc. So these blue-green algae increase the soil fertility by adding organic matter to the soil. So that's all about biofertilizer, where the microbes which helps in the biofertilizers. Students, in this video we discussed about the sewage treatment. So here we study about in brief primary treatment and the secondary treatment and importance of the BOD. And next we study about the production of biogas. So there we were studied by the step process which involved. Okay, the mechanism of the productions we studied in this session. Thank you.